Seems to be my name. Well, I want to thank the organizers for including me in this great meeting. It's always a pleasure coming to Italy, eating Italian food, drinking great wine like we had last night. And you had nice weather yesterday. I'm not sure about today, but and so I'm going to extend this concept about type 1 interferons and anti-tumor immunity. But in order to get there, I have to tell you where we started with this project, which is analyzing human melanoma metastases to try and understand mechanisms of clinical response versus failed response to immunotherapies such as vaccines. And what we found was that at baseline, there's a subset of patients, about a third of melanoma patients, that has a pre-existing smoldering immune response against their tumor that is characterized by chemokines that can mediate T-cell recruitment. These are chemokines associated with T-cell markers such as CD8. And it seems that it's these patients who are responding preferentially to immunotherapies. Not everyone, but the responders fall into that subset. And so it turns out that seems true preliminarily with our vaccine approaches, but also with anti-CTLA-4 antibody. There was a small biomarker study published by the BMS group of about 60 melanoma patients treated with anti-CTLA-4 antibody. And then in the early days with anti-PD-1 antibody also, it seems to be this subset of patients with pre-existing smoldering immune response against their tumor that's doing something, but obviously it hasn't rejected the tumor because the tumor is still there. And what we noticed when we were looking for innate immune signs, signals that might be mediating this perhaps upstream from the T-cell response, this subset of patients has a type 1 interferon signature in their tumor microenvironment. The mechanism of immune escape here is presence of immune inhibitory pathways such as PD-L1. I'll show you some of this data in a minute. On the other hand, in melanoma, it's about 2/3 of patients that completely lack this phenotype. No CD8 T cells, no type 1 interferon signature, no PD-L1, IDO, FOXP3, no negative regulators. And so, and these patients are probably resistant to our current immunotherapy. So I think a major hurdle for the field is to figure out what we can do with these patients who have a non-T-cell inflamed tumor in order to capture them for clinical response to immunotherapies. And I think we have some clues about how to do this now that we understand the role of the type 1 interferon pathway in the natural immune response. And we've summarized this in a recent review article, which you can read if you're interested. But these T-cell infiltrated tumors probably have an activated vasculature that's getting the T-cells in collaboration with those chemokines, but the T-cells are shut off by negative regulation. In contrast, these non-infiltrated tumors lack chemokines, probably don't have an activated vasculature, and we actually consider this subset now to be sort of immune exclusionary, where you can have circulating T cells against tumor antigens, but they're not going into the tumor microenvironment. So we have to figure out ways to open that tumor microenvironment, get the innate immune inflammation going, and get the T cells there. So a few points about this T cell infiltrated tumor microenvironment. What we have observed is that these are the tumors that have upregulated expression of PD-L1 that engages PD-1, the inhibitory receptor in activated T cells. These are the tumors that have upregulated IDO, the tryptophan catabolizing enzyme that's been shown to be involved in peripheral tolerance, and also Tregs. And we have evidence I won't be able to discuss today about T cell intrinsic anergy also being a key negative regulator. So this presented us with a paradox because we had imagined that the immune suppression in the tumor microenvironment was set up by the tumor so that whenever T cells came in, they would be shut off. But these results suggest the opposite, that maybe the T cells are responsible for upregulation of that negative regulation. So that turns out to be the case. These are now immunohistochemical stains for CD8, FOXP3, PD-L1, IDO. You can see an example of a T cell infiltrated tumor that has expression of PD-L1 IDO and it has FOXP3 Tregs. Without CD8 T cells, we generally don't see that phenotype. We have statistical correlations with CD8 and FOXP3, and also between CD8 and IDO, or CD8 and PD-L1. And the mechanism of this then depends on the CD8 T cells, and we modeled this in mice. So with B16 melanoma, for example, in vitro, it doesn't express IDO. It has low expression of PD-L1. if we put the tumor in vivo in immune-competent mice, let the tumor grow for a week, take it out, and look for IDO transcripts. We have some experiments with protein now. It's induced in vivo, but that's completely ablated in gamma interferon knockout mice or CD8 knockout mice, CD8 deficient mice. So you need gamma interferon and CD8 T cells to upregulate IDO. Same thing for PD-L1. You have low PD-L1 expression in vitro, you put the tumor in vivo, let it grow for a week, harvest it, throw it on the fax, it's now upregulated PD-L1, but that too completely depends on gamma interferon and CD8s in the host. So in a way, this is a negative aspect of gamma interferon. It's absolutely necessary for tumor rejection when we see it. If we use gamma interferon knockout T cells, there's no tumor rejection. But on the other hand, it's also responsible for upregulation of these two inhibitory pathways that in a chronic immune response serve as a kind of an immune intrinsic negative feedback loop. Same thing for Tregs. Tregs, if we do adoptive transfer systems to pick apart migration into the tumor, Tregs also preferentially migrate into the tumor only when we transfer CD8 positive T cells only. This is not gamma interferon dependent, it's mostly mediated by chemokines made by the activated CD8s. And so this T cell infiltrated, tumor microenvironment is mediated by chemokines, innate immune inflammation, the activated T cells migrate in, and then if antigen positive cells are not completely cleared, the negative feedback shuts down the rest of the reaction. So we have just a couple points on therapy related to these observations. We have therapies that are being tested in the clinic. Anti-CTLA-4 antibody is already FDA approved, but therapies blocking PD-1, PD-L1 interactions, trying to deplete Tregs by targeting CD25 mostly, IDO small molecule inhibitors, and strategies to try and reverse T-cell intrinsic energy. I'll just show a couple of slides from the early clinical trials if you're not actively following this literature, but it's actually quite impressive, the clinical benefit that's seen in a subset of patients, and it seems to be the subset of patients that has this pre-existing T-cell infiltrate. These are tumor measurement curves in individual patients with just anti-PD-1 antibody alone, without any predictive biomarker. There's no patient selection, and the response rate is about 30% in those patients. And combinations are being pursued to try and double up and block two inhibitory pathways together. I myself have a number of clinical trials open or in development with combinations with anti-PD-1 and IDO inhibitors, and some of the published data from anti-CTLA-4 plus anti-PD-1 already look quite impressive. So I went through that a little bit fast, but I needed to sort of set up the problem that I think with these current data in the next couple of years, I'm pretty convinced we're going to have great immunotherapies for the patients that have that pre-existing smoldering immune response. So what are we going to do with the rest of the patients, right? And here I think we have to get the innate immune recognition going again. So in order to pursue that line of investigation, we wanted to first understand what is the natural mechanism of innate immune recognition of tumors when it does occur, and then maybe we could try and mimic or bypass, sort of reproduce that natural effect. And so this was alluded to earlier. We and Bob Schreiber's lab as well reported in 2011 now, late 2011, that the natural T-cell response that occurs in vivo in mice depends on host type 1 interferon signaling. And we proved using a variety of knockout mouse strategies, mixed bone marrow chimeras strategies, conditional knockout, that first of all, very quickly after you implant a tumor in vivo, you detect beta interferon largely, but also alpha interferon transcripts from CD11C positive cells. And that type 1 interferon production has to act at the level of the CD8 alpha DC. And if you either have CD8 alpha DCs absent in the mice, or if you selectively delete the type 1 interferon receptor in the CD8 alpha DCs, you lose everything downstream. So you absolutely need this type 1 interferon response to the tumor in vivo in order to get the natural T-cell response. So that's moved the question upstream. What is it about the tumor that's inducing beta interferon production by dendritic cells? And that's something we think that we've just figured out. If you think about candidate pathways that could lead to beta interferon gene transcription, You have TLR pathways, cytosolic RNA sensing and cytosolic DNA sensing pathways. So by putting tumors in different knockout mice lacking these different components of innate immune signaling, we used MyD88 knockout, TRIF knockout, specific TLR knockout mice saw no defect in the T cell response against the tumor. We used IPS1 knockout or MAVS knockout mice to look at the RNA sensing pathway. There was no defect in the T cell response against the tumor. And so that left us with the STING pathway. And it turns out that, that if we use STING knockout mice or IRF3 knockout mice, of course downstream from STING, we completely lose the T cell response against the tumor. And immunogenic tumors that are normally rejected spontaneously, you completely lose the tumor rejection. And this seems to be relatively unique to the tumor context. If we put skin grafts into STING knockout mice, for example, they're rejected normally. So it's something about the tumor that's different from another tissue like an organ. And during the process of this recognition, we've done, I think, fairly sophisticated experiments to prove that we find tumor-derived DNA in the dendritic cells in the tumor microenvironment, just like you have protein sort of being cross-presented by those dendritic cells. And so we've put this together. It's a paper that was just submitted strongly suggesting that it's tumor-derived DNA, perhaps from dying cells that are turning over, that depends on the CGAS, STING, TBK1, IRF3 pathway. The dendritic cells that result are really well activated, and all of this is necessary for the T cell response against the tumor and then subsequent tumor rejection. And this is based on knockouts of STING, IRF3 in vivo. So another point that came up earlier, I put in an extra slide here because the question has come up about transplantable tumors and endogenous retroviruses, and that many of the tumor cell lines that we use in the laboratory have endogenous retroviruses that are expressed or made. And so we've worked this into a genetic tumor model, and I want to tell you about this a little bit. In the clinic then, if you think about it in terms of population biology, there is a question of interpatient heterogeneity. There are some patients that make this natural T cell response against their tumor like this example, right? This is a CD8 stain. This tumor is full of T cells. It's a lung metastasis, right? How do the T cells get there, but not in this patient's tumor, right? And so this could be germline polymorphisms, perhaps. It could be somatic differences at the level of the tumor, or environmental differences, such as the gut microbiome that Giorgio talked about earlier. And we have data suggesting that all three of these can be important. And what I want to do is show you just one of these, which is somatic differences at the level of the tumor, where what we've been doing is exome sequencing, RNA-seq, gene expression, profiling, to try and identify oncogene pathways that are on in these non-inflamed tumors that might be excluding T cells. And so we've identified 4 candidate pathways, and what we're doing now is we're engineering those into inducible transgenic mice. So this is a tamoxifen-regulated CRE. In our case, it's driven by the tyrosinase promoter, which is on in melanocytes. And the conditional transgenic or conditional knockouts are bred into the mice. The mice develop fine. And you smear a little tamoxifen on the skin, and within 3 weeks they get a melanoma in that spot, driven by those recombined oncogenes. And so when we do that, the driver model here is active BRAF, which is a very common driver mutation in melanoma, and then P10 conditional knockout. When we do that, you get melanomas. They start to arise in 3 weeks. 100% of the mice, they go on to be 100% fatal. These tumors do have a natural T cell infiltrate. So that tells us one thing: you don't need a transplantable tumor, poking a needle through the skin of mice, injected under the skin in order to get a T cell infiltrate in the tumor. Now we've engineered onto this model other oncogenes, and without going into the details, we have one oncogene that we co-express by breeding onto another conditional transgene. These mice get melanoma at the same time frame, no T cell infiltrate. Right? And when we look at the beta interferon, well, here are T cells in the tumor. This is with just BRAF P10. The T cells that are there co-express LAG3 and PD1. They have this sort of dysfunctional phenotype. When we sort them out, we stimulate them, they're poor at cytokine production, just like the human tumors and just like the transplantable tumors. But when we add this third oncogene, we don't get any of those T cells. And this is what I wanted to show to support beta interferon production. Once those tumors develop, if we separate the tumor cells from the host cells, we can easily detect beta interferon transcripts in the CD45 positive cells from those tumors. So again, that's with no transplantation of a tumor, no injection with a needle through the skin. This is a genetic tumor, arising relatively in these mice. And this happens with some oncogene combinations and not other oncogene combinations. It's the same mice, same mouse facility, litter mates, same microbiome. The only difference is that extra oncogene. So I think we're sort of convinced away from the argument that this is an artifact of transplantable tumors and not just retroviruses. And therapy, this is just like the clinic. So in the T-cell infiltrated melanoma oncogene permutation, you give anti-CTLA-4, anti-PD-L1 antibodies, a few doses, we slow the tumor growth with this indication. In the other model, where there's no baseline T-cell infiltrate, no therapeutic effect, and no T-cells migrating into those tumors. These are innate immune strategies in a genetic tumor model where there's no spontaneous immune response. So can we take a completely cold tumor, non-inflamed at all, and convert it into something that can support T-cells? So what are those innate immune strategies? So the two things I'm going to show you here, STING agonists to try and mimic that STING pathway, which is driving type 1 interferon production and other things from dendritic cells, and then giving type 1 interferons themselves, but into the tumor microenvironment. And here we have some interesting results depending on how the type 1 interferons are given. So a few years ago, there was a drug that was developed called DMXAA that had an unknown mechanism of action. It had therapeutic effects in mice. It was moved all the way to phase 3 clinical trials in cancer patients in man. That was published a couple of years ago. That was completely negative, that trial. But only last year was it realized that this drug is a STING agonist, and it turns out it binds and activates mouse STING, but not human STING. Right? So now, armed with this information, we're re-exploring its therapeutic effect, the mechanism of action, and we're working with a company to develop new agonists that bind human STING. And so this drug, in B16 melanoma, you can establish it for 7 days, for 14 days, a single injection into the tumor, complete tumor disappearance in almost all of the mice. It's the most powerful immune therapy that we've ever seen in any model. Anti-CTLA-4 antibody, anti-PD-1. When we've used a beta interferon reporter mouse so that when the beta interferon gene is activated, we detect YFP, I think it is in these mice. Within very short time after injection of DMXAA, you see this very large beta interferon signal by cells in the tumor microenvironment and in the tumor draining lymph node. When you gate on those interferon beta producing cells now and do a FACS, they're almost all CD11C positive cells. This is CD11C by F480. So you get the response that you want, and it's a huge response. This is an order of magnitude above the endogenous beta interferon response just by putting in the tumor. When you look at T cell responses against tumor antigens, here I've plotted this as individual mice, just to see the ELISpot assays. It's 20-fold higher, this T cell response, than you get with the endogenous, and higher than we've gotten with any other therapeutic intervention. And the mice, once they reject the tumor, they're completely protected against challenge. It's long-term memory. It's an extremely impressive phenomenon. So we're working then on STING agonists for the human to mimic this response, and we're plugging this into the non-inflamed genetic melanoma model to see if we can really convert a completely non-inflamed tumor microenvironment into one that's inflamed, supports T-cell activation, chemokine production, and then migration of those T-cells back into the tumor at the end. And so what about interferons themselves? And we had done experiments in mice with systemic alpha interferon or beta interferon. They both seem to work. They have a therapeutic effect, and there's a modest therapeutic effect. But if the mechanism, the immunologic mechanism of type 1 interferons is through activating the CD8 dendritic cell compartment in the tumor draining lymph node, and we want that to be focused attention just on that compartment, then maybe intratumoral administration of type 1 interferons makes more sense. So this is something we've explored. And then something that was alluded to earlier, we've thought about ways to target type 1 interferons to the tumor microenvironment, and this we've done with single-chain antibodies against HER2 or EGFR. And then if we have therapeutic effects, we've been working out the mechanism, and the mechanisms of these things are slightly different, and I'll explain that. So this is intratumoral type 1 interferons. We've done this injecting multiple doses of alpha interferon or beta interferon. The cheap experiment is to transfect the tumors to make their own beta interferon or alpha interferon. You know, major therapeutic effect, there's no doubt about it. The tumors regress. We've done this in a number of ways. Alpha and beta both work. I think beta is a little bit better. Intratumoral is better than systemic. And a depot, you know, keeping it going seems to be better than just one dose, right? But what's the mechanism of this? So it turns out that that mechanism, this is a high dose of interferon, type 1 interferons in the tumor, right? Turns out that mechanism is completely non-immunologic. It works in T-cell deficient mice, it works in NK cell depleted mice. It turns out that when we did bone marrow chimeras, that this effect depends on type 1 interferon signaling on non-hematopoietic cells. And since we're injecting it into the tumor, it has to be some stromal cell in the tumor. And so this is well described with type 1 interferons, the anti-angiogenic effect. Turns out, you know, when you do this kind of experiment, you completely lose the vasculature in the tumor. This is, an analysis of the neovascularization in the tumor microenvironment. And then what we did was we obtained Tie2 Cree mice crossed to the conditional type 1 interferon receptor knockout to get rid of type 1 interferon signaling selectively in endothelial cells. And then you lose most of the therapeutic effect with that too. So with high doses of type 1 interferons in the tumor, we're convinced this is completely anti-angiogenic. And maybe high dose interferon in the clinic in melanoma, maybe that's anti-angiogenic too. What about low doses of interferon? So I told you already, the endogenous low doses of type 1 interferons made in response to the tumor are priming the T-cell response, and you need that low dose to get the T-cell response. So how can we get low doses of beta interferon into the tumor microenvironment? So the strategy that we worked on with Yang-Xin Fu at the University of Chicago is making single-chain monoclonal antibody constructs coupled to beta interferon. And we've done this with anti-EGFR and with anti-HER2, and this is very potent, right? So this is with an EGFR expressing tumor with anti-EGFR alone, or the interferon anti-EGFR conjugate, and this is the tumor control that you get. This is completely T-cell dependent. If you deplete T-cells, you lose this effect. And then we've done the conditional knockout experiment also, deleting the type 1 interferon receptor from different cell lineages. And if you delete it from dendritic cells now, this is a CD11C Cree to get rid of type 1 interferon signaling in dendritic cells, you completely lose the therapeutic effect. So this is more in trans, where the antibody is binding tumor cells, but the effect of the type 1 interferons has to occur on the dendritic cells. And this was recently published if you want to take a look at more detail. So the major points I wanted to make here are that this T-cell inflamed tumor microenvironment, I think, is a pretty good candidate as a predictive biomarker for response to our current immunotherapies that are really gaining traction in the clinic. Some vaccines, anti-CTLA-4 antibody, anti-PD-1 antibody, maybe the combinations. That when that spontaneous immune response does occur, I think all of our data from the human correlates in melanoma, from the transplantable tumor models, and now from the genetic tumor model, oncogene-induced model, suggests that the mechanism of this spontaneous immune response depends on the type 1 interferon pathway, and in the transplantable tumors, it completely depends on the STING pathway. We don't know yet in the genetic tumor model. So with that as a backdrop, then, we think that STING agonists could be a really fantastic approach to explore clinically. This has to be done now with new molecules that bind and engage human STING. And targeting type 1 interferons to the tumor microenvironment, I think, is also a reasonable clinical strategy. And the mechanism of therapeutic effect may very well depend on the dose and schedule of that type of interference that you introduce. So I need to call out in particular a few people who've contributed to this work. Yuan Zha in my lab, and now she runs our immune monitoring core facility, is driving this melanoma genomics interrogation to look for molecular mechanisms of the T-cell infiltrate in human tumors. Mercedes Fuertes completely drove that paper and project describing the role of host type 1 interferons in the natural immune response against tumor, and Robert Spapin did the introduction of type 1 interferons into the tumor. The genetic melanoma models have largely been driven by Stephanie Spranger, and we have a lot more data on this point that I didn't get to show you. And then the innate immune sensing and STING pathway is being worked out by Seng-Yong Wu, a Korean postdoc in the lab, and Leticia Corrales, a Spanish postdoc in the lab. And from there, I'll take questions. And I spoke really fast so that there would be 2 minutes left. So, thanks.
Thank you. You are perfectly on time, so there are time for 2 questions, please.
Hi, I have a couple of questions. First one is, have you noticed other immunosuppressive cells infiltrating the T-cell inflamed melanomas, apart from the T-regulatory cells? I mean, have you looked at myeloid-derived suppressor cells or and2 macrophages?
Yeah, so maybe let's take it one bite at a time. Yeah, so in all the tumors, there are some kind of myeloid cells, right, that are probably suppressive. In all the tumors, there's TGF-beta, IL-10. Some of the T-cell infiltrated tumors have the highest level of TGF-beta and IL-10. There doesn't seem to be much correlation with clinical outcome. It's sort of built into the biology of growing a solid tumor, I think. And so we hadn't focused on that as a primary mechanism. We also see some of the transcripts you associate with MDSCs like arginase. We see that expression in a subset of non-T-cell infiltrated melanomas, so I don't think it's limiting the function of T-cells that are there. So I'm not saying that they're not important, and as we work out the mechanisms, especially of how some of the oncogene pathways are completely excluding the host response, some of that may end up being through myeloid cells. But.
Yeah, so you think that is the tumor that shapes CD8 T cells to induce the recruitment of these suppressor cells? I mean, how do you think?
Yeah, so for the Tregs, for the FOXP3 positive Tregs, it's dependent on the CD8s. And we look in...
And why do the CD8s do that inside the tumor and not maybe in other contexts?
That they do. So if you look at multiple disease scenarios with chronic inflammation, you have Tregs that fall on come in behind the effector cells. Remember that there was a New England Journal paper of kidney transplantation, and groups have been looking at transcripts in the urine correlated that might predict rejection. And the best transcript that was identified was FOXP3. Because as you get inflammation with effector cells, the Tregs follow. And there are multiple examples like this, yeah?
Very short, please.
So you mentioned very quickly about the melanoma model, the activation of STING by cyclic GAS. So do you think other DNA sensors can activate the pathway in other tumors, or?
So far we've done 3 transplantable tumors, and all of them depend on STING. And maybe we'll find one that depends on something else, but right now that seems to be the focal point. We just are getting the CGAS knockout mice. We've been working a lot with that molecule as the ultimate sensor. In vitro, everything is CGAS dependent, so we'll see. We'll see in a few months. Yeah.
Last one. Filippo, very briefly. Yes.
Well, most of the observations that you made in tumor models resemble some data that we obtained. Like you mentioned, that, for instance, when we use interferon beta focused on tumor cells, it was really an anti-angiogenic effect that turned out to be very important. What about this STING agonist that you used in your experiment? Is this developed at the clinical level? It's very promising.
Yeah, so the STING agonist in mice, it doesn't work in STING knockout mice, and you lose most of the effect in T-cell deficient mice. And so most of that, but there's a little bit of leftover therapeutic activity. Most of that is immune. Some of that might be innate, right? So we need to work out the rest of it. For the clinic, like I said, we're working with one company for human agonists. They're already identified. We put them in the mice. They work the same. We stimulate human cells in vitro, and these stimulate human cells to make beta interferon in vitro, whereas DMXA doesn't. I'm hoping, optimistically, 2 years. These are cheap to make, right? This isn't like making, an antibody, right? It's a small molecule. So hopefully it'll go into the